Well, we're going to continue with muscles. A very, very brief review since the last video. Here you have skeletal muscle. You have a muscle fiber or a muscle cell, as it's accurate to say both of those. Around the outside of the muscle cell is a tissue called a sarcolemma. This word here, sarcolemma. Inside is a material called sarcoplasm. It's the same thing as cytoplasm. It's just inside a muscle cell, so we call it a sarcoplasm instead of cytoplasm. Same stuff. Inside one muscle cell are a whole bunch of these units called myofibrils, and myofibrils are made of back-to-back-to-back structures, all right, that are known as sarcomeres. So a sarcomere goes from that Z line to this Z line. Sarcomere. The big red thing in the middle, that's a thick filament. The thinner little blue things are thin filaments. Thin filaments are composed of 3 different proteins that you have to know about. There's actually more proteins than that, but there's 3 you have to know about. Thick filament is composed of a protein called myosin. All right, here's, remember, muscle cells or muscle fibers are multinucleated. There's a nucleus here. There's a nucleus here. If you keep on going down the length of the muscle fiber, you're going to find more and more nuclei. Lots of mitochondria. Mitochondria everywhere. This sucker needs tons of ATP to function. So here are the terms you need to use. Remember, they're multinucleated. The sarcolemma is the plasma membrane around a muscle cell. Sarcoplasm is the fluid, the equivalent of cytoplasm inside the cell. Many, many, many myofibrils inside one muscle cell, or one muscle fiber, as this is. Right, myofibers have thin filaments and thick filaments. They're composed of sarcomeres. Those are the individual units that are back to each other. Surrounding the myofibrils is this structure called a sarcolemma. I'm sorry, sarcoplasmic reticulum here. And then in between units of the sarcoplasmic reticulum, they are bordered by this thing called a T-tubule, or that stands for transverse tubule. So we have this structure here called a triad. A triad consists of this. This is exactly what a triad is. 1 transverse tubule and the 2 sarcoplasmic reticulum cisternae. That's the ends here. That's this part and this part on either end. That structure, those 3 things, are called a triad. And it says that right there, triad. All right. Moving along, same slideshow here. Next slide. So here is a close-up view. We've got more of a close-up view here. This is the sarcolemma, right? So this is the skeletal muscle fiber or muscle cell. There's the sarcolemma on the outside. Here's a myofibril. We've taken one myofibril and pulled it way out. We pull that myofibril out, and you can see the thick and thin filaments alternating. You can see this is a sarcomere. That's a sarcomere. From here to here is another sarcomere, and then we've taken it and we look at the view that it looks like under a microscope slide. And all the zones that we've talked about before. Here are the zones you need to know about. I band, A band, H zone, Z line, and M line. And they're all outlined here. And we've talked about those in previous views. Here's the sarcoplasmic reticulum. This would be one end of it. That's a cisternae. The cisternae is the end of a sarcoplasmic reticulum. It ends into a T-tubule there. And then on the other side of that is the other sarcoplasmic reticulum, cisternae. So these 3 things together are called the triad. This is known as striated muscle because it has that banded appearance. That banded appearance is only found in skeletal muscle and in cardiac muscle. So here's the bands. Again, the I band. Make sure that you outline what a sarcomere is. A sarcomere is defined as Z line, and that's this dark line here. That dark line, that's a Z line to this Z line. That is a sarcomere, one sarcomere. Inside the sarcomere, you have these other bands that we identify. So sarcomere, Z line to Z line. right in the middle, this line that they don't have any label on, that line right in the middle, that red line that goes up and down right there, that's called the M line. That's what this is. That anchors the thick filament. And it's in the center of the A band. The A band is defined as the length of a thick filament. So the length of one thick filament is there to there. The length of the thick filament is from here to here. That corresponds to what we call the A band. All right? The lighter area that's in the middle of the A band is called the H zone. That's from here to there. That's the H zone. And then the I band is the light bits on either side of the Z line. Sometimes the Z line is known as the Z disc. You might see it referred to as that. Same thing. If it's Z, it's Z. All right, so now we're going to zoom way in. We zoom way in. Here's myosin. This is the thick filament. This is the middle of the thick filament right there, where on the previous slide you would find the M line. Right, so it goes from out there to out there. And it continues on. You can't see the rest of it. Okay, so that's the thick filament. The thin filament is up here. The thin filament has 3 proteins that you need to know about. Here are those 3. Actin is the primary one. You probably already heard of that. This little ball-like structure, one little ball-like structure, is an actin globule. So we put a bunch of actin molecules together, we get part of the thin filament. Another molecule that you need to be aware of is this one called troponin, or troponin. So troponin is this molecule, this interesting looking, looks like a rocket ship in there. That's the kicker, that's the key molecule, key protein that will help the muscle to contract. The third protein you need to be aware of is this one here called tropomyosin. Tropomyosin is the long banded, long skinny protein that sits inside, looks like it sits inside a groove on the actin chain. So here's the 3. Actin is the little ball-like one. You got lots of those all strung together. Troponin is this smaller blue globule right there. And then the tropomyosin is the long string-like protein that sits inside the groove. It is actually covering up what are known as active sites in the actin. We're going to get to that. All right, so contraction of a muscle fiber. The contraction is defined as the Z line, from one Z line of the sarcomere to the other Z line of the sarcomere. When they get closer together, that's called a muscle contraction. But here's how they get closer together. Get closer together by the movement within the myofibril. The myofibril shortens. Actin and myosin filaments slide past one another. That is known as the sliding filament theory. The sarcomeres shorten. That is if one sarcomere shortens, you get a little bit of a pull. If you get a million sarcomeres that are all back to back to back and they all pull a little bit, you get a much more dramatic contraction of the muscle. All right, now, how do you contract the muscle? So this involves the nervous system. I've mentioned before how muscles really are inactive. Really, they don't do anything until they're told to do something. They're really good at doing what they're told to do, but they do nothing unless they are told. And the thing that tells them what to do, when to contract, is a nerve. And in the particular case of skeletal muscle, this striated muscle, in the particular case there, it is a motor neuron. This thing right here called a motor neuron. This is the axon of a motor neuron. Remember, the axon delivers a message from the cell body, which would be up here somewhere. The cell body sends a message. It's called an action potential. We're going to get deep into that in the next lecture. So this electrical signal comes down the motor axon, down to here, to the end. The end of it is called the neural bulb, sometimes referred to as the axon terminal. There are lots of different names for this, but where the axon ends, it meets up with the outside of the muscle cell membrane. So remember, the muscle cell membrane is called a sarcolemma. where the motor axon meets the sarcolemma. Where that meets, that whole structure right there is called this. That is what a neuromuscular junction is. And you can break it down. Neuro means it has something to do with the nervous system, right? Muscular has something to do with the muscular system. Here we are. And it's the junction between those two. So the neuromuscular junction consists of the end of a motor axon where those meet with the sarcolemma of the muscle. That's a neuromuscular junction. Again, as I said, skeletal muscle fibers only contract when stimulated by a motor neuron. Motor neuron. That means movement, right? We're creating movement. There are two big kinds of neurons in your body. There are sensory neurons. They feel things. They send messages back to your brain. We'll get deep into that. They send messages back to your brain that you're feeling something, that something is going on out there in the world. This is not that. This is the other kind. This is the motor neuron. This is the neuron that is telling this muscle, do something. I want you to do something. Contraction is what it's going to do, but it's telling it to do it. Otherwise, if the motor neuron is silent, the skeletal muscle fiber is also silent. It doesn't do anything. So here's the parts of a neuromuscular junction. So that's the shorthand, NMJ, neuromuscular junction. You've got a motor neuron. There it is, the motor neuron. You have the motor end plate. So the motor end plate is considered the part of the muscle fiber, the sarcolemma, where the end of the neuron, the end of the axon meets. So that is considered this bit right here, where on the sarcolemma, where the terminal end of the axon meets. We consider the motor end plate as only part of the muscle fiber, not a part of the nervous system. All right. Then you have a synaptic cleft. A cleft. What's a cleft? A cleft is a gap. So, yes, the motor axon meets at the sarcolemma of the muscle, but it doesn't touch it. There's a gap there. That gap is called a cleft. You see that here, synaptic cleft. It's very, very tiny cleft, very tiny gap, but it's there. At the end of the axon terminal are what are known as synaptic vesicles. Inside the synaptic vesicles, those little ball-like things, inside there are neurotransmitters. That is the chemical that will cross the gap and will attach itself onto the sarcolemma, and the sarcolemma will then react, knowing that this is the signal to go ahead and contract. But there's a whole process involved in getting the muscle to contract. And then, of course, you have to have a neurotransmitter. The neurotransmitter is the neural chemical that will cross the cleft and attach to the sarcolemma to tell the muscle, go ahead, contract. All right, that particular neurotransmitter in the skeletal muscle system is called acetylcholine. Acetylcholine, always abbreviated capital A, capital C, small h. Acetylcholine. Anytime you see ACH, they're talking about acetylcholine in the skeletal muscle system. That is the neurotransmitter that is going to cross that synaptic cleft, attach to the sarcolemma, and cause that muscle to do its thing, to contract. But we got to go through the process. It's a complicated biochemical process. So it's the nerve impulse that causes the release of acetylcholine. You have to have a nerve impulse come down here. When the nerve impulse comes down here, it will cause the neurotransmitter to be released in a process that we have to go through as well. The acetylcholine binds to the acetylcholine receptor. So the receptor is on the motor end plate. The receptor is on the muscle cell, on the muscle cell membrane. Right? That acetylcholine will cross over. It will attach to the acetylcholine receptor that is specific, very specific for acetylcholine. And then it will cause a change in the motor end plate on the muscle cell. Here's the change. Acetylcholine causes changes in the membrane permeability to sodium and potassium. Remember these ions. So remember the membrane is pretty much a nonpolar entity for the most part. It acts as a nonpolar barrier. That means things that have charge to them find it very difficult to go through that membrane. Acetylcholine causes a change in that membrane permeability. So they allow channels to open up to allow sodium and potassium to flow through the membrane. So that's called changing the membrane permeability. When that happens, that generates what's called a muscle impulse, or it's called an action potential. Now in the nerve, that is definitely what it's called, an action potential. But you have to now start to think of muscles as you think of nerves, right? These are electrical creatures that need an electrical activation of some kind in order to contract. Now there's two ways that you can cause a muscle to contract. Of course, you can tell your own motor neurons to release acetylcholine into the synaptic cleft, and they bind to the acetylcholine receptor, and that causes a depolarization of the muscle cell membrane, and that causes the muscle to contract. Great, that's one way to do it. You can do it yourself. Or I can take an electrical device like an electric stimulator that I use in physical therapy, and I can attach that to your skin, and then I can send electrical impulses into your skin. And if I send enough electricity, I will cause your muscles to depolarize and contract for you. You may not like it when I do that, but that's a way that we can help people that have a certain degree of paralysis, to help them to stimulate their muscle fibers so they don't get smaller and atrophy to too large a degree. That impulse, that acetylcholine, causes the release of calcium from the sarcoplasmic reticulum. And I told you in the earlier video, sarcoplasmic reticulum is a calcium storehouse. That's what it's doing. It keeps calcium in there. When it's told to release it, it lets it go. When it lets it go onto the muscle contracts. Again, there's a specific pathway for that to happen, and specific events have to happen for that to be the case. All right, so let's continue on. Here we go. All right, so this has a name. The name, I'm going back here, the name of the combination of the nerve telling the muscle what to do and the muscle doing it. That has a name. The name for that is what's called excitation contraction coupling. Okay, that means the excitation comes from the nerve. The nerve is telling the muscle. The neurotransmitter crosses the synapse. It depolarizes the muscle cell membrane, leads to the release of calcium. The calcium binds to certain proteins, which we're going to get to in depth. That causes the muscle to contract. That is a coupling. If you do not have excitation of the muscle cell membrane, you do not get contraction. Let me repeat that again. If you do not get excitation from the outside of the muscle, you do not get contraction. So if you slice the nerves going to any skeletal muscle, you do not get an excitation going to those muscles, and those muscles do not contract when you tell them to. No nerve, no muscle contraction. If you have a nerve, you can't get a muscle contraction. So that's where you get the excitation contraction coupling. All right? So there's a definition. The connection between muscle fiber stimulation and muscle contraction. All right. During muscular relaxation, the calcium ions are pulled back away from the molecule that they have to attach to, and I'm going to get into that deeply, that deep connection. The calcium ions are stored in the SR, sarcoplasmic reticulum. Burn that into your mind. Sarcoplasmic reticulum. It's a huge long word. Nobody likes to type it out over and over again. SR stands for sarcoplasmic reticulum. All right. Now, let me show you pictures that you have in your own book. These are the pictures that come from your own book, right? So here's the myofibril. This is all one muscle fiber, that whole thing, or a muscle cell. One muscle cell has the sarcolemma. There's the sarcolemma peeled back away, showing you all these myofibrils, tons and tons of myofibrils. You have sarcoplasmic reticulum in blue. I can zoom in on that a little bit more. Watch this. Okay, so yes, this is available in your book. So there's sarcoplasmic reticulum, there's sarcoplasmic reticulum holding calcium, and then at the endpoints, there's the sarcoplasmic reticulum, there's the green T-tubule, and then there's the other sarcoplasmic reticulum. You remember that's called a triad. That's the triad. That's where most of the calcium is stored, inside there, okay? Here's a nucleus. Remember, it's multinucleated. You got lots of nuclei. All right, let's go back to regular view. So now we're going to move. I'm going to go past this one. That's a little bit complicated just yet. Now we're going to talk about the contractions here. We're going to review those proteins again. Here's myosin, right from your textbook, myosin. Here's all the head groups on myosin. It's the head groups that do the work of myosin. That's a thick filament. And it's thick filament. Look at that, 11 nanometers. I mean, that's not real thick, but 11 nanometers. Over here, 5 to 6 nanometers, much thinner. Of course, this is the thin filament. The thin filament has 3 proteins that I want you to know about, okay? The 3 proteins are actin, the red one that looks like a ball, strung like a bead of pearls, string of pearls, all along that line. Okay, on actin, there is this little, I'm gonna zoom in a little bit closer. On actin, see that that little area, darker colored area on the actin molecule. Okay, that's called an active site, or it's what's known as, what they say here, a myosin binding site. That should tell you something. Myosin wants to bind to actin right there. But look what's in the way. Something's in the way. That thing that's in the way is called tropomyosin. Tropomyosin blocks the binding sites that myosin wants to grab onto on the actin. Can't get to them. If you can't get to it, you can't get a muscle contraction. That's tropomyosin's job, is to block that. This molecule's job, troponin's job is to act as a gatekeeper. It will either allow tropomyosin to stay there, to stay put, and block the myosin binding site, or it will move tropomyosin out of the way under certain circumstances. And those certain circumstances are when calcium is around. When you have calcium around, and normally when the muscle is not contracting, you don't have calcium around. When the excitation comes from the motor neuron stimulates the sarcolemma depolarizes. When that depolarization takes place, the sarcoplasmic reticulum that had the calcium in it lets go of the calcium. The calcium is now free to bind to troponin right there. It binds to troponin in that little shallow groove. That changes the conformation of troponin. Troponin pulls tropomyosin out of the way. Now, the binding sites for myosin are freely available to the myosin head groups. Now, these myosin head groups now grab onto the actin, to the binding site that they want so bad on actin. So, then we can start the actual muscle contraction from that point on. So here's the pathway, right? Upon muscle stimulation, right? You have to have the motor neuron causes transmittance of acetylcholine across the synaptic cleft onto the muscle cell membrane. Muscle cell membrane impulses, that means you're spreading a wave of what's called a depolarization. A depolarization wave causes the sarcoplasmic reticulum to release calcium ions into the cytosol. That means into the sarcoplasm, right? The fluid that is bathing the myofibril. Calcium binds to troponin and changes its shape. The shape change causes tropomyosin to be pulled out of the way. The change in the shape of troponin alters the position of tropomyosin. Now the binding sites on actin that are specific for myosin are exposed. The binding sites are exposed. The myosin heads bind to actin, forming cross bridges. Right? That's a term you need to know. Cross bridge. A cross bridge is when myosin and actin are connected to each other. That's called a cross bridge. That only happens when calcium is present